Today we will be transfecting 4T1 cells obtained from ATCC. We will be using a transfection kit manufactured by Altogen Biosystems.
In this tutorial, we use a 24-well plate. Transfections can be scaled up or down based on plate surface area as described in the corresponding transfection protocol, which can be found at altogen.com. Start by seeding 500 microliters of cells in a complete growth medium into the wells. Cell density should be at 50,000 cells per milliliter. Incubate the cells for 12 to 24 hours before transfection at 37 degrees Celsius in a humidified carbon dioxide incubator. When you are ready to transfect, aspirate the growth medium, wash the cells once with PBS, and add 500 microliters of fresh growth medium. To prepare transfection complex, mix 40 microliters of serum-free medium, 5.5 microliters of transfection reagent, and either 750 nanogram of DNA or 30 to 50 nanomolar siRNA or microRNA. An optional step is to add 2 microliters of complex condenser, which increases transfection efficiency, but may also increase cytotoxicity. Incubate the prepared transfection complexes at room temperature for 15 to 30 minutes. Add the prepared transfection complexes to the 500 microliters of complete growth medium. Once you have treated all appropriate samples, incubate the cells at 37 degrees Celsius in a humidified carbon dioxide incubator. Assay for phenotype or target gene expression 48 to 72 hours post-transfection. Here is a Western blot validation of our transfection reagents. 4T1 cells were transfected with either cyclophilin B DNA, which resulted in an overexpression as compared to non-treated cells, or cyclophilin B siRNA, which resulted in partial silencing. Thanks for watching our tutorial. For more information or products, visit us at altogen.com.